Hi everyone. So in the last video we talked about the process of aligning your RNA-seq reads and kind of some of the post-processing steps that you have to go through um, after you've aligned your reads. And in this kind of, not the last video in this series, but my last video for right now, we're going to talk about the process of assembly, which is a little bit different than alignment. We talked briefly about that difference in the last video, but I'll kind of go through it one more time here just to zoom in and refresh on alignment and what that looks like. We're taking, I can actually just draw it down here. This is assembly. So if I have my reference genome, which I know this is very short, it would be many, many times longer than this. When I align, I just take my individual reads and I place them where they match along this stretch of sequence. And hopefully I don't have a lot of big gaps unless there are actual gaps in the transcriptome, but it's impossible to know if a gap like this is because I just didn't happen to get a read that placed there properly for whatever reason, or if this is actually a gap where we would not see RNA being transcribed. But again, in alignment, we're just lining up all of our RNA-seq reads in terms of where they fit on this reference genome. For assembly, we take this information in blue and we create kind of comprehensive transcripts of what we actually think the sequence is and kind of the unbroken sequence based on where these reads align. So your data will look a little bit different after this, but it is largely based on the alignment and just kind of contains a little bit more of a cohesive idea of the transcript, whereas the alignment data is still very fragmented. It's just where do these reads fall, not really what does this cohesive transcript look like. So this process of going from the alignments over to the assembly, kind of this step right here, is what we're doing with a program like StringTie. And StringTie is the main assembler that I saw and wanted to use. There is an assembler called Trinity. And Trinity can actually be used without a reference genome. So it uses De Bruijn graphs, which we can talk about in another video, to kind of go straight from these reads right here, which I've circled here, which would actually not be assembled with a reference genome. They would just be assembled based on overlaps between the reads, kind of piecing them back together. It would go from these reads to a complete transcript, but without a reference genome, potentially. That's another option, but StringTie worked amazingly for me. I really loved it. It was super easy to use, so I would really strongly recommend it. So it's going to take your BAM files that you generated in HiSat2 as input. So you take your BAM files, and you'll actually input them one at a time, to my understanding. So I had BAM files from different tissue samples. I had some from the head, some from muscles, and I fed each individual one in to StringTie. And then I got a GTF file as output, which again, we can take a look more thoroughly at some of these file types, and I can make a video on that at another time. But it's just another way of representing kind of the sequences and where it aligns on the genome. So I would input my BAM file, I would get a GTF file out for each tissue. So for each tissue, I repeated this process and generated, for me, it was 7 separate GTF files. And there is then a setting called merge on StringTie, where you can feed in all of these GTF files and you actually get like a merged GTF at the end based on all of them. So it gives you a little bit of a better idea of the transcriptome as a whole. But these individual GTFs that are generated by tissue type are going to tell you, again, right, what is being transcribed in each tissue. It's going to be a little bit more specific, but it's nice to also create the merged GTF because then you're going to end up with kind of just a little bit, you just have more data, and more data never really hurts. So it's nice to look at both and compare them, I would say, looking between kind of what is suggested by the tissue specific assembly versus what is suggested by the merged assembly. So you'll generate these files, and then there's an option in StringTie called merge, where you can feed in those GTFs and get your end product. So after that, you officially have an assembled transcriptome. So that was kind of start to finish the process that I initially indicated that I wanted to do right up here, where I said I want to go from my RNA-seq data all the way over to transcriptome assembly. So after StringTie, we have officially assembled a transcriptome. But as I've kind of hinted at throughout the video, kind of all of these file types are very difficult to look at on their own just because there's so much data. So I would say your work isn't really necessarily done in terms of assembly after StringTie. My very last step for assembly itself would be using IGV or something similar, which this is for visualizing your data. And visualizing your data is really important because, again, these are not things that you can just look at the raw data and really get a good idea of what's going on. If you want to look at the structure of genes, etc., you're going to need to visualize it. And IGV does a great job of that. It is a desktop application. You can also run it in the command line. And you feed in your reference genome that you've been using throughout all of these steps. You can also feed in the BAM files from HiSat2. So you can look at how do my reads align, and that can be really, really helpful to understand maybe where exons and introns are and the different kind of splice variations. It's really helpful to see that. It also gives you an idea of the transcription, how frequently something is transcribed, because you can visually see how much it's transcribed. And you can also input the GTF files from StringTie, which again is kind of giving you then that overall look at, based on the reads and the alignment, what do I think that the transcripts might look like. Now, I will note for y'all, because this really stumped me, you don't just need a BAM file, you also need something called a BAI file, which is a BAM index. And you can create this using our good old trusty samtools.index. And then you just input your BAM file and it spits out this .bai file type. And it needs to have, weirdly enough, like the exact same name as the BAM file. Otherwise, IGV doesn't recognize it. So IGV has some fun quirks like that, where you do need kind of these index files. I think you might also need a FASTA index file for your reference genome, which you can either generate with BLAST, I think has a featured index, but IGV can also generate the index itself. So you do need some index files. The GTF is loaded in totally fine. It's like a dream. You just click and it's there. But the other two files can be a little bit more of kind of wrestling to deal with getting them up on the screen. And there's great guides to IGV, though. So it has really, really great documentation. I would say all of the tools that I mentioned here have really great documentation, which is really helpful when you're just starting out in terms of your kind of bioinformatics learning process. Okay, so once you've visualized, you're kind of at an okay pausing point. The next steps that you could go through, the two kind of big routes that I have seen are either quantification of transcription, so understanding quantif, that was not spelled right at all. Let's try that again. Quantification. There we go. Quantification. So how much are these genes transcribed? This can be really helpful with like differential expression analysis, where you're saying, okay, I'm looking at RNA sequencing data from these two different samples, and I want to understand if they express certain genes at different rates. So quantification is a really important step. I've been looking at using ball gown for this, which is a pretty user-friendly program that can be run in R. If you're familiar with R, it's really nice to go back to using R, because a lot of the rest of these programs are not compatible with R from what I've seen. So Ballgown is really nice in that sense. But there are there's also different bioconductor packages that can do this kind of quantification step. If you're more interested in purely the expression of these different reads that you're looking at. But there's also a path that is more looking at gene prediction. So if you're working in more of a model organism, which is something that I am doing, you might not know a lot about what the actual transcriptome or genome looks like. That's kind of the state that I'm at. So you might be interested in taking these or this assembly information, which, kind of maybe gives you some vague ideas of where genes are, um, but not perfectly, because, like, for example, a gap like this, it could either be an intron, right? So maybe these are just two exons in one gene. This is exon 1, well, exon 1, exon 2. Or, on the other hand, it could be an actual gap in between genes. And maybe this is gene 1 and maybe this is gene 2, right? So we don't know what that gap indicates. So gene prediction models can help with that, looking at those sequences and understanding whether or not they're part of the same gene, where the splicing is occurring, et cetera, et cetera. So gene prediction is a great next step as well, and more kind of along the lines of what I'm doing. Breaker and Helixer are two gene prediction models that I will be using in the near future. So I'll be able to make some videos on gene prediction once I've actually gone through that process and I have a little better idea of how it works. There is some pre-processing before gene prediction. For quantification, at least for ball gown, you can actually generate ball gown files via string tie. So they're, I think, part of kind of the same like suite of programs. So string tie can create files that are compatible with ballgown 2, and ballgown 2, or yeah, ballgown 2 is like very easy to use. So that's fantastic. But I'll make more videos on both of those processes probably in the next weeks as I'm learning and successfully, hopefully, doing those processes.